All right, engineers, in this video, we're going to continue on with the discussion, and we finished up with the late distal tubule, right? And then we're getting ready to talk about the intercalated A cells and B cells and then the collecting duct. All right, so that's what we're going to talk about now. We're going to finish off, and we're going to discuss the activity of the intercalated A cells, intercalated B cells, and the actual antidiuretic hormone acting on the principal cells. And even this would also be, remember I told you this is a special type of cell? This cell right here is actually called a principal cell. So let's actually write this down. This is called a principal cell. And principal cells are just basically cells that maintain your actual mineral balance and your water balance, whereas these intercalated A cells and B cells are maintaining your acid-base balance. But again, if you guys remember where we left off, we talked about sodium chloride reabsorption within the early distal tubule via the sodium chloride symporters. We talked about calcium reabsorption, which is dependent upon the parathyroid hormone. And we talked about in the late distal tubule how aldosterone can actually cause increased sodium reabsorption and cause potassium ion secretion. And then also we said if the antidiuretic hormone is present, it can actually increase the expression of aquaporins, aquaporin 2 specifically, right? And then that will actually allow for water to flow in and follow the sodium ions into the blood, which would not only increase sodium and decrease potassium levels, but it would increase the water, which would increase your blood volume and increase your blood pressure. Okay, so now that we've done that, let's go ahead and see how these suckers are working here. Okay, so let's do the first one, which is the intercalated. We'll do this one, we'll say this is the intercalated A cell. And I'm gonna kind of underline that A cell because this is really going to be easy to remember why we have intercalated A cells and B cells. Thank goodness whoever named these darn things, they were kind to us and helped us to make it a lot easier instead of giving someone's name to it. You know, thank goodness for that. Okay, so intercalated A cell and intercalated B cell. These are the two ones that we're going to talk about quickly. Intercalated A cell, remember this is for acidosis. So A is for the acidosis type of conditions. So respiratory acidosis, metabolic acidosis, and then the B is for basic conditions. So basic conditions, what do I mean? Alkalosis. So alkalosis. Okay, so whether this be metabolic alkalosis or whether this be respiratory alkalosis. This is how our body can compensate for that acid-base balance and help to be able to keep it within the homeostatic range. So let's see how it does it. It's really cool. Okay, so let's do the A cell first. So let's say that you actually have high CO2 levels within the blood. So if the CO2 levels actually kind of move out of the blood and they'll go into our actual intercalated A cells. When they come into the intercalated A cells, this CO2 will combine with what's called water. And in the presence of this enzyme here, it'll form, so what's this enzyme here called? This enzyme is actually really important. Let's write this enzyme down. This enzyme is called carbonic anhydrase. And carbonic anhydrase is actually stimulating this step. And what it's doing is it's confusing the water in the CO2 and converting it into what's called carbonic acid. So H2CO3. And this is called carbonic acid. Then what happens is carbonic acid dissociates into two things. One of the things is protons. The other thing is going to be bicarbonate. Okay? So two things it dissociates into. One is going to be the protons and the other one is going to be the bicarbonate. Okay, well, let's think about what the problem was. If it's acidosis, what does that mean that the pH is? That means that in acidosis you have a low pH. That means that there's a lot of protons, okay? A lot of protons within the blood. Okay, cool. That means that there's very little bases to be able to counteract these protons. All right, well, this is a base. This is bicarbonate. Bicarbonate is a good base. He's a weak base, but he'll be able to tie up some of those protons. Well, that means I want to get a lot of this base into the blood to tie up those protons. And I don't want any more protons in the blood because it's making the pH acidic. Let me get rid of these protons. So that's what our body does. And look what it does. Here's this channel here. Let's put this channel protein right here. So look, here's this nice channel protein. And this channel protein, what it's going to be doing is, remember there were some of these potassium ions that were being excreted here? These potassium ions are sneaky. They're like, hey man, you want to get rid of me? You can't get rid of me. I'm coming back at you. So what happens? Some of these potassium ions are going to try to come into the cell. And what happens is some of these actual protons are going to try to go out of the cell here too. And because you're moving protons from actually low concentration to high concentration, and you're moving potassium ions from low concentration to high concentration, what do you think this requires? This requires ATP. So this will require ATP. So this is actually going to be a ATP-dependent pathway. Okay. Now, you know what else can actually happen here too whenever you're in acidosis? Sometimes in certain situations, your body wants to be able to secrete certain substances that it doesn't really like. What are some of those substances that it doesn't really like that it can actually deal with? One of them is ammonia. So you see here NH3, this is called ammonia. This stuff is toxic, man. Super neurotoxic. It can cause a lot of problems. Cerebral edema, a lot of different things. So what can happen is ammonia has specialized transporters here within the luminal membrane. And whenever ammonia is actually brought in, look what can happen to this ammonia. This ammonia can actually be excreted out. If this ammonia is excreted out into the urine, look what can happen here. These actual protons that you pumped out can combine with the actual ammonia. So now this proton can actually combine with this actual ammonia here. And when these two combine, you're going to get what's called ammonium, which is a weak acid. Okay? And this is one of our actual buffer systems within the urine. Okay, cool. So that's one mechanism right there. Okay, well, we still didn't take care of the issue. We got rid of some of the protons. That's outstanding. But what about this bicarb? All right, well, let me fix this carbonic anhydrase, and let's just denote it for right now. Since we know that enzyme, let's denote it Ca, since we can make some room here. So Ca is carbonic anhydrase. Now look what happens. This bicarbonate has channels here in this actual basolateral membrane. And on this basolateral membrane, I'm going to pump the actual bicarb out. And then where can this bicarb go? I can take this bicarb and I can put this bicarb into the bloodstream. And as I increase the bicarb in the bloodstream, what happens to the pH in the bloodstream? What eventually happens to the pH? The pH will go back up to normal levels because some of those bicarbonate ions will tie up some of those protons. But you know, like anything, we have to have things being transported with it. So there's a specialized transporter right here because you're losing negative ions, right? So we have to bring negative ions in. So we bring chloride ions in to prevent that excessive negative ion shift. Okay, so that's how the intercalated A cells are working. What about the intercalated B cells? Take this pathway and flip it now. Instead of getting rid of the protons, get rid of the bicarb. And instead of actually putting the bicarb into the blood, put the protons into the blood. That's all we're going to do. So let's see how that works. And we're going to move through it fast because we already know how it's going to work. So again, if there's CO2 here and there's water, what can happen? These two guys can fuse. If these two guys fuse, you'll form carbonic acid. Carbonic acid can actually dissociate into protons and into bicarbonate. And again, what was this enzyme here that was controlling this step here between CO2 to carbonic acid with the water here? This was the carbonic anhydrase. He was catalyzing this step here. And then what was happening? Okay, well now we have this channel here. Let's have this channel here. And now what's going to happen is we're going to take this bicarb and we're going to get rid of that bicarb. But again, we're losing negative ions. So what ions would I want to come into the cell? I'd want there to be negative ions coming into the cell to compensate for this negative ion loss. So guess who comes to the rescue? Chloride. And chloride can come into the cell. And then there's special chloride channels here on the basolateral membrane that the chloride will actually exit through. Okay, and it'll leave out of the cell. Okay, that's cool. So I got rid of some of this bicarb. I'm urinating out a lot of the bicarb in this situation. Okay, so I'm losing a lot of my base. All right, cool. And again, why would I be doing this? Why would I be activating this pathway? It's the exact opposite of this one. So what does that mean for the pH? Okay, well this pH must be really high. I must have a high pH. That must mean that I actually have what? A lot of bicarb in the blood. And I have not a lot of protons in the blood. Okay, where in this situation you had high amounts of protons and very little bicarbonate. And we fixed that by increasing the bicarb in the blood and getting rid of the protons. Now we're just going to do the exact opposite. We're going to reabsorb the protons and get rid of the bicarb. So that's how we do it through this pathway. So that will get rid of the bicarb this way. But then we have to bring these actual protons in. So now there's going to be channels to bring the protons in. And whenever you're pushing the protons against that gradient, you're going to want to bring the actual potassium ions in here. And so some of these potassium ions can be brought into the cell. And because both of these guys are going against their concentration gradient, this pathway will require the presence of ATP. It's an ATP-independent process. Then, If these protons that you're actually bringing into the actual blood come into the blood, what happens to the pH? The pH goes back down to homeostatic range. Okay? So that's how the intercalated A cells are working, and that's how the intercalated B cells are working. You know, another thing that your actual your collecting duct could actually do besides that, you also have other cells that can be secreting certain types of substances like drugs and toxins. So drugs, toxins, and even another breakdown product called creatinine. Or creatinine, I should say it like that, creatinine. So it can secrete toxins, can secrete drugs, can secrete creatinine. And what else did we secrete? We also secreted ammonia. And we can also secrete protons. And we can also secrete bicarb. So that's actually showing us some of the secretory processes and some of the actual reabsorptive processes. But now we have one more thing left to do. Okay. Now let's say that we have to release this other hormone. And we talked about it a little bit. We said that there was this ADH, right? Well, let's go over here to this structure. And again, this is not the testicles. I know it looks like it, but this is the actual hypothalamus, and this is the pituitary gland. Now, in the pituitary gland, remember here in the hypothalamus, before we say that, remember in the hypothalamus we had the collection of these neurons here? And these neurons were actually coming specifically from the supraoptic nucleus. So these actual cell bodies up here in the hypothalamus, what is this again? Supraoptic nucleus. And then they had these axons that actually, what? Move through from the hypothalamus to the posterior pituitary. And whenever it was stimulated, it would release the hormone, antidiuretic hormone. And again, what else do we call it? Vasopressin. Now the question is, why are we releasing vasopressin? What's the stimulus? Why are we doing this? Because that's what we want to ask ourselves. We always want to ask ourselves, why is this happening? Not just knowing, oh, this just happens. Why? Okay, so ADH is going to be released whenever the plasma osmolality is changing. Okay, so what do I mean by that? All right, before I do that, let me explain what he's doing, because that's also going to help us. So osmolality. Okay, so before I even explain whether it's high or low, let me explain what ADH does real quickly. ADH pulls water from the kidney tubules into the blood. If it pulls water into the blood, remember the terms, I want you guys to remember the term hypertonic, hypotonic, and isotonic. Hypertonic means that there's more solutes than there is water. Hypotonic means that there's more water than solutes. Isotonic means that there's actually equal amounts of solute and water. ADH wants to pull water into the bloodstream, so it wants to have more water in the blood. What does that mean then? That must mean that the actual blood was hypertonic originally. There wasn't a lot of water. So the plasma osmolality must be high. Okay? And that's why he must be being stimulated. What's another reason why he can be stimulated? Another reason why is because of this sucker angiotensin II. He just loves to stimulate everybody. So angiotensin II can also stimulate ADH. So angiotensin II can stimulate ADH as well as a high plasma osmolality. And even certain drugs and other stimuli can stimulate the ADH production. But we're going to stick with these two main ones. Increased plasma osmolality, which means there's very little water and high amounts of solutes. And the second one is that there's angiotensin II production, meaning that you want to increase your blood pressure. Okay, well, how is it going to do all this crap? Let's see. Okay, so ADH is released. ADH comes over here, and he sees his receptor, and he's like, oh, yeah. What is he going to do to this receptor? Okay, let's actually show this receptor right here. Here's this receptor. Here's our vasopressin receptor, right? This is our vasopressin receptor. And whenever it actually binds on to this vasopressin receptor, what happens? ADH binds onto this receptor on the actual kidneys, specifically in the collecting duct, because now we're talking about the collecting duct. So again, intercalated A cell, intercalated B cell, they can be found not just in the collecting duct, but they can also be found in the late distal tubule. Okay, so intercalated A cells, intercalated B cells are found in the actual late distal tubule and in the collecting duct. And this last one here, what is this type of cell? This is another principal cell, because he's controlling our water and ion balance. This is the one that ADH is acting on. When he acts on this vasopressin receptor, he'll activate the G stimulatory protein. He's bound to, who binds to GTP, becomes active. As he becomes active, he comes and activates another effector enzyme. This effector enzyme is called adenylate cyclase. When it activates adenylate cyclase converts ATP into cyclic AMP, which then goes and activates protein kinase A. What does protein kinase A do? I'm so glad you asked. So here inside of this cell, you're going to have these special vesicles, these pre-synthesized vesicles. Look at these vesicles. They're so cool because they have on them these specialized channels. These blue protein channels here, these blue protein channels are aquaporins, but they're a special type of aquaporin. These aquaporins are called aquaporin 2. And why am I telling you that? Because if you guys have watched our endocrinology videos, you'll know that there's actually aquaporins 3 and 4 on the basolateral membrane. So what are these ones? Aquaporin 3 or aquaporin 4. So you guys would know that now, right? Now, another thing is that there's actually going to be these proteins that are present on this vesicle. They're like basically snare proteins. And what protein kinase A does is he comes over here and he puts some phosphates on these different proteins. By phosphorylating this, it causes this sucker to start fusing with the actual membrane. When this thing fuses with the cell membrane, then what happens? Well, now let's actually show you here. Look, there's going to be protein channels. I'll draw all 3 protein channels here. And again, what are these protein channels? All right? So these protein channels here are going to be aquaporin 2. So this is aquaporin 2 right here. Originally, there was no channel there. There was no channel there. But then when ADH came to this area, what happened? It activated this whole second messenger system and did what? Led to the fusion of these vesicles with these aquaporin 2s to the membrane. Now, water is going to start flowing through these channels from areas of high concentration to low concentration. Where will this water go? It'll go through here and then out through the aquaporin 3 and 4. Then through the aquaporin 3 and 4, where can that water go? It can go into the bloodstream. As water goes into the bloodstream, what happens to the blood volume? The blood volume goes up. So as water comes into the blood, an increasing amount of water, our blood volume goes up. What happens to the blood pressure? That goes up. So that takes care of the initial stimulus, which was the angiotensin II. But what about more water? Well, if I have more water, I'm going to have my water increase to where it can become equal to about where my actual solutes is. So that takes care of my plasma osmolality. So it brings the plasma osmolality down till it reaches a normal point. So it actually reaches a normal plasma osmolality. It tries to make it isotonic, right? So about 300 milliosmoles. Okay, that takes care of that issue. And again, if you remember, I just want you guys to make sure that you understand this. We talked about this guy here, ADH. He can work in the collecting duct and he can work within the late part of the distal tubule. Okay? So if the ADH and the aldosterone is present, it can reabsorb some sodium and water. And that is actually variable. So remember I said here, 10% of the sodium was remaining, right? Out of that 10%, 5 to 6% of it was actually reabsorbed here. What about that remaining 4 to 5%? The remaining 4 to 5% is right here. This is the remaining 4 to 5%. So the remaining 4 to 5% is actually going to be dependent upon the presence of aldosterone. Calcium was also getting reabsorbed here, but it was dependent upon the presence of PTH. Then what did we say? 20% of the water was being reabsorbed. Okay, well, what does the water depend upon? Oh, well, we said water is dependent upon these aquaporin 2s. Well, again, what was this one here? Aquaporin 2. This would be aquaporin 2, right? And this is dependent upon ADH, right? ADH has to stimulate the aquaporin 2 formation, right, on the luminal membrane. And it's not just happening here within the, again, you can call this like the late distal tubule or the cortical collecting duct. And then this is actually going to be more of your actual later part of the collecting duct. So again, Antidiuretic hormone can act at the late distal tubule all the way down throughout the collecting duct. So if ADH is present, it can pull water. But the amount of water that's being reabsorbed, that percentage of water, is variable because it depends upon the body's demands and it depends upon the presence of ADH. If you have a lot of ADH, more water reabsorption. Less ADH, less water reabsorption. Simple as that, right? Okay. Now, one more thing here. You guys remember here within the Vasa Recta? Within the Vasa Recta, what was this again? Let's go back down our actual medullary gradient. Okay, this was 300 milliosmoles, 500 milliosmoles, 700 milliosmoles, 900 milliosmoles, 1200 milliosmoles, right? So it's getting saltier as you move down. Why? Remember, it was because of this countercurrent multiplier mechanism. As the ascending limb is going up, it's pumping sodium, potassium, chlorides, calcium, magnesium out, which is pulling water out of the descending limb. The Vasa Recta, again, what is this structure here called? This structure is called the Vasa Recta, and the Vasa Recta is just basically a paratubular capillary network present within the deep part of the medulla. And what it's doing is, as you're going down, it's getting saltier. Who would want to come out? Water. Water wants to come out to where it's salty, right? Because it loves to go out to where the salty areas is, that obligatory water movement, right? Okay, well, in the same time, as you're going down, salt is actually going to move from high concentration to low concentration. So some of the sodium chloride is actually going to move in here. And then as I have some of this sodium chloride moving in, why am I doing this? Okay, well, hold on before I explain why I'm doing it in a second. As we turn back around, the process reverses. So now water is going to go back into this actual capillary network. And as water tries to go back into the capillary network, that's kind of interesting. All right. What happens to sodium chloride? The sodium chloride gets pushed out as it's going back up. Why in the heck is it doing this? If you guys remember, what did we call the Vasa Recta? We called this a specialized thing. We called it the counter current exchanger, right? And we said that the countercurrent exchanger of the Vasa Recta had two functions. One is prevented the rapid removal of salt. That was one of the big things. We said that it actually did two things, right? One is it prevented rapid sodium chloride removal. That was one of the big things because as it's going down, it's actually pulling in some of the sodium chloride from the medullary interstitial space and providing a little bit of water onto that area. But if this is actually happening so much, if we actually have so much water reabsorption, it can wash away a lot of the salt. So we don't want to lose a lot of that salt from the water reabsorption. So what do? We pull some of the salt into this Vasa Recta because it's coming in at 300 milliosmoles. But when it leaves, the actual the osmolarity of the blood coming out of the Vasa Recta is approximately 325 milliosmoles. So it actually takes a little bit more sodium chloride with it to prevent the rapid removal of the salt from the medullary interstitial space. What else did it do? It also provides oxygen to the nearby tissue cells, right? But it's a very sluggish blood flow, we said. Why am I telling you this? Because remember we said that this was contributing to the medullary interstitial gradient, which was trying to pull the water out, make very, very concentrated urine. That's the whole purpose. There's another thing that's contributing to this that's also helping in this process. His name is urea. So urea is also contributing to this process. Look here. So urea is right here. Urea actually gets reabsorbed in the last part of the actual collecting duct. So in the last part of the collecting duct, after a lot of the water is being reabsorbed, the urea concentration here starts really increasing. And by facilitated diffusion, it moves out of the collecting duct and here into this medullary interstitial space. As it's going down, look what actually happens. It actually gets reabsorbed back here into the actual ascending limb. But as it's doing that, urea actually accumulates out here. And urea is a solute. If urea is accumulating out here for a little bit before it actually gets reabsorbed, what is it helping to do? It's helping to make the medulla even more salty. So this process here is called urea recycling because it just keeps going. It'll actually go back in here and then go back and get reabsorbed. Now some of it will actually get lost in the urine. Let's not confuse that. A lot of it will get lost in the urine, but some of it will actually get recycled. This is called urea Recycling. And again, this is important because why? It actually is helping to maintain, it's actually helping to do two things. Make concentrated urine. That's what it wants to do. It wants to make concentrated urine, and it wants to contribute to the actual medullary interstitial gradient. So it wants to contribute to medullary gradients. All right? To be able to make concentrated urine, in other words. Okay. So we covered, what have we covered before we actually end this whole video, guys? We covered within this video, we talked about the intercalated A cells and how they work in acidosis. We talked about the intercalated B cells and how they work in alkalosis. We talked about the principal cells responding to the antidiuretic hormone and how that actually helps to increase water reabsorption. We talked about how the collecting duct and the distal tubule are also playing a role in secreting drugs and toxins and creatinine and protons and even ammonia. We discussed a little bit about the countercurrent exchanger and how that's helping to prevent the rapid removal of salt and provide oxygen to the tissue cells nearby. And we talked about how this urea recycling is helping to contribute to the medullary interstitial gradient, and it's helping to make concentrated urine. Ninja Nerds, I hope all of this made sense. I really hope you guys enjoyed it. I really hope it did. In the next video, what we're going to try to do is we're going to try to take everything and put it all together and see how all the reabsorptive and secretory mechanisms are actually occurring, so that you guys can see everything in a nice one action flow, because that's what we really want to do. We want to make everything make sense. All right, Ninja Nerds, so we're going to have one more video, and that's going to be the entire overview of the entire reabsorptive and secretory processes. All right, Ninja Nerds, I hope it made sense. If it did, please hit the like button, subscribe, put some comments down in the comment section. As always, Ninja Nerds, until next time.